Hi, everyone. My name is Davon Crawford. I am a birth and postpartum doula and a pre-nursing student. Um, and for my future career, I plan on going into midwifery services or midwifery practice. I plan to practice midwifery and basically work in my local community. Um, and so presenting the material that I'm presenting to you today is very near and dear to me. And so I want to say thank you for allowing this space and for listening to my presentation. And today I will be presenting on 16S rRNA. And the title of my presentation is How 16S rRNA Can Be Used to Reduce Maternal Disparities in African American Women. I hope you enjoy my presentation. So a little background on maternal disparities in the African American community is that African American women have higher rates of infant mortality, preterm, and adverse birth outcomes compared to all of the other major racial and ethnicity groups in the United States. And so at this point, we know it has a lot to do with, racial oppression, systematic oppression, and, a lot of it currently has to do with the climate that we live in today, as far as, the social and economic climate that we live in today. But nonetheless, research is finding that not only does maternal and fetal environment play a factor in maternal outcomes for African American women, but the microbiome of African American women also play a role in gestation. And so I wanted to present this to you today because with the longstanding nature of this disparity or the disparities, science can now suggest the need to shift from microbiological forces that shape disease risk in particular communities. And so this is the reason why I chose to do 16S rRNA, because hopefully it can continue to make some change in the community. Okay, so what does 16S rRNA target, and how is the technology used to help patient outcomes? So 16S rRNA, the gene is present in all bacteria, including eukaryotes. And since the variable region of 16S rRNA provides specific species signature sequences, it can be used to target pathogens in patient specimens. And so this technology can be used to help patient outcomes because the 16S rRNA PCR has the ability to detect and identify certain and particular bacteria pathogens, and therefore can reduce some of the maternal disparities as it relates to microbiome diversity. And of course, specifically help kind of change the disparities that African American and other underserved communities currently face. So we'll get into how does 16S rRNA work. So the 16S rRNA gene encodes the molecules of ribosomes that are responsible for the essential process of converting genetic messages to functional cell components via the translation of mRNA to proteins. And let's see. I don't want to give you too much. I'm going to take up a lot of time if I read everything on my slides, but I'll read a little bit more. 16S rRNA gene sequencing is used for identification, classification, and quantitation of microbes with complex biological mixtures in environmental samples, such as marine water, vaginal, and gut samples. And so it is a highly conserved component of the transcriptional machinery of all DNA-based life forms, and is highly suited as a target gene for sequencing DNA in samples containing up to 1,000 different species. Okay. And so now, what will hospitals and clinics need to do to successfully offer 16S rRNA sequencing? In medical microbiology, 16S rRNA sequencing serves as a rapid and cheap alternative to phenotypic methods of bacterial identification. And because 16S rRNA gene sequence analysis is more sensitive when detecting environmental samples, it becomes a useful tool for environmental surveys in hospitals and healthcare clinics in distinguishing HAIs or with population-specific studies that may require vaginal microbiome samples of those at risk for preterm birth. So in order for hospitals or clinics to successfully offer 16S rRNA, variations of technology is needed to support testing, such as thermal cycler, gradient PCR machines, PCR workstations, and qPCR machines. So like I said, this is a technology that could potentially change maternal outcomes and just kind of distinguish and establish certain bacteria that may kind of have run rampant in a hospital in different ways like that. So I hope that you enjoyed my presentation. I have another page here with my references. And hopefully, as I continue into my nursing degree and into maternal health, some of these testing and more studies and hopefully 16S rRNA sequencing becomes a more popular subject, and we can continue to make changes in not only the African American community, but in all underserved and in women's studies in general. So thank you for listening. I hope you enjoyed it. Take care.